Good evening. So I'm going to go through lab 3. I decided to make a PowerPoint with all the pictures that we would have saw during this lab together. So I will be putting a recording up so you can watch it over and over again. As always with the lab book, I would focus on those vocab words here. We'll be using them a lot throughout the semester. And you need to be responsible for knowing those terms for your lab practical. So in this lab, we would have taken the skills that we would have learned using the microscope, and we would have then looked at some actual cells. Now remember, we're studying two different types of cells. We're talking about either prokaryotic or eukaryotic. Pro meaning before the, pro means before, and karyon means nucleus, so before the nucleus. So this would be our bacterial cells. And then eukaryotic after the nucleus. So you need to know the difference between pro and eukaryotic cells. So I kind of talk about this in a couple different places throughout this presentation. So first of all, we would have looked at a really cool organism called Oscillatoria. Now, Oscillatoria, this is a pre-made slide, so all you had to do is put it on your microscope, find it, and look at it. Now, these are prokaryotic cells. So these are like bacterial cells that when they divide, when they go through cell division, they secrete this mucus, and so they stay attached. And so as they divide and replicate, you get these long filaments. Now, do notice they are green. They are green because they do have chlorophyll. You talk about chlorophyll. They do photosynthesize, but what they do not have is they do not have chloroplast. They do not have membrane-bound organelles. So they have a specialized membrane called a thylakoid membrane that that is where photosynthesis occurs. And again, if you do the reading, you will find this. So I highly suggest that you read through the lab book and answer the questions. So then in the questions, it asks you to draw the specimen. And so we have a picture here to include much detail as possible. And then it says, based on your what you observed with your microscope, can they be measured accurately? And so the next slide I have is a picture here of Oscillatoria. And this one's really magnified, but we can see the individual cells here. And so we could kind of accurately, with what we know with our field of view and how big our field of view is in the different objectives, which is what we learned on the last lab. So we know that at a 40x objective, our field of view is 0.45 millimeters. And so we can count how many cells go across it and divide it by that number. So we take 0.45 and then divide it by the number of cells, and that would give you how many cells. And then I did ask if it was photosynthetic, and I really already answered that question. So that would be the end of the Oscillatoria. And then we move on to yogurt. And this one's actually kind of a frustrating one if all the cells, because the bacteria are really small. All right, so we would have taken a sample of yogurt, which we kind of dilute down with some water, and you would have made what's called a wet mount. Do make sure you watch the video on how to make a wet mount. We would have then stained it with methylene blue, and Professor Hayes shows you how to move, or what I call it wicking. When you pull things through using that paper towel, we would have wicked the methylene blue underneath the cover slip, and we would have then been able to see the different bacterial cells, which we can see in this slide in our yogurt. And so then you're going to answer the different questions that you would have on it. It shows you on page 26 how to make that wet mount, and then you would have made a drawing. And then the question is, can you accurately measure this? Well, these cells are so small, it would really be difficult to get an accurate measurement of these particular cells. And then it asks, is a nucleus visible? Well, again, remember, prokaryotic cells do not have membrane-bound organelles. So you know, a lot of students will say, oh, they're too small. No, they don't have them. They are bacterial cells. Remember, they're prokaryotic, before the nucleus. So you're looking for structure they just do not have. And then we move from prokaryotic onto eukaryotic. So when we start with our eukaryotic, we're going to talk about plant cells. Now with these plant cells, you can see the nice cell walls. Also, we notice that plant cells tend to be boxy or rectangular in shape. You can see the nice chloroplast inside there. And the chloroplast will actually move around the central vacuole. The central vacuole is full of water. It's in the middle, so you're not going to see it. It's not stained. So they'll move around. That's called cytoplasmic streaming. And we do have a video for you to watch on that phenomena. And then here's what the plant would look like. So it's an aquarium plant called Elodea that we would have looked at. So you would have pulled a leaf off of it, put it on a slide, and then you would have seen all these neat things. So here you can see that empty space here. It's really not empty, it's full of water. That's your central vacuole, your chloroplast, and your cell wall. And then here is a picture, because it does ask you to draw, and then it asks you if you can tell the size. Well, this is at 100x, and we really only use the 40x. So this is a little bit bigger, but you can count how many cells go across the field of view. So this one you could actually get somewhat accurate measurement. And then we would have seen this. This is a already made slide we would have looked at. And these, what they're showing you is they're called plasmodesmata. And so we have here like a cartoon little diagram. And since plants are multicellular, they do need to have a way of communicating from cell to cell. And with that cell wall, it kind of gets in the way. And so what's going to happen is we're going to make these little tunnels from cell to cell, and those are called plasmodesmata. And so we can see those. This next slide is actually very similar to what you would have saw under the microscope. And you've got a, you can see the little, like I said, they look like little straws going between the cells. And so then it would have asked you some questions about the specimen and the function. So it's asking you to look at its structure, and then it's asking you for what's its function. What does it do? And then we would have moved to procedure 3.5, and these are the onion cells. We would have made a wet mount with this. We would have used neutral red in order to stain some of the organelles inside. Again, onions are plants, so we see our cells are kind of box-like. We also see the nice cell walls, and then we see the nice nucleus in each of the cells. Now, if you look carefully, you can kind of see it here and here and here. You can actually see, and I have one blown up. Inside the nucleus, you have these dense little circles here as well, and those are called the nucleoli, and we'll talk about the function of the nucleoli in our chapter material. And then it asks you to, again, to draw in your notebook here and to label those structures. And then we would have moved on from an onion to a potato. And what I like to do with the potato is that you kind of make a scraping, and then what we can see is with the iodine complexes with starch, and you get this black color, and that actually looks like stained glass windows. It's one of the more prettier slides we do. And so we can see the starch granules inside. Here's our cell wall material. This structure is called an amnioplast, and the amnioplasts are in the family with the chloroplasts, except for the function. Instead of harvesting solar power to make food, which is what many of your plastids is another name for these types of structures, this particular plastid's job is to store extra food, starch, in plants. So of course, you know, potatoes are all full of starch. So that would be your amnioplast. So you can see that there. It asks you to draw that, and so we would have our cell wall and our starch granules. And then we're going to move on to the cheek cells, and you would have used your own cheek. You would have taken a toothpick, you would have gently rubbed the inside of your mouth, and you would have swirled it around in a drop of water on your slide, put the cover slip on, and then wick through the methylene blue again to get nice stained cheek cells. Now notice the shape of the cells are different. Animal cells tend to be more circular. You should also notice there's not a defined cell wall because, again, animal cells do not have them. But what we can see with the methylene blue is we can see the nice nucleus stained in the middle of our cell. And then if we look even closer, and I have another picture here, and this one's nice and labeled for you, you can also see on the surface of your cheek cells, there are bacterial cells. And so we can see that. A lot of times it shows you the cytoplasmic organelles. So a lot of times what we're seeing is the mitochondria, and those are kind of look like it's been peppered in there. Usually if we use a different stain like Janus green, we'll get a better picture of those mitochondria, but you kind of can see those organelles. There's something else inside that cell, and that's full of organelles. And so again, you're supposed to draw this. You can, if we go back, this would probably be about the view you would get under your 40x, maybe a little bit bigger, maybe more of 100x. But you can see, you would be able to approximate the size. So I don't have an actual one that you could do, but you are supposed to do that on your virtual microscope. So that is that 5-point extra assignment there, is to go to that virtual microscope. And again, the one with the Adobe Flash Player has a cheek cell to look at. The other one does not. It had like I think a sand dollar larvae. So pick one of those specimens in there. Give me a screenshot and tell me what you think the approximate size of that cell is for those 5 points. And then we move into procedure 3.8, which are protists. Again, Kingdom Protista has a lot of different types of organisms that live in there. Some are single cellular, some are multicellular. We have tiny things from what we're going to look at, the amoebas and the paramecium, to huge kelp. Okay, kelp seaweed are also in this kingdom. So we have, those are multicellular. And then we have some really cool things called Volvox. Volvox is colonial. I don't happen to have one of those on PowerPoint, but if you look up Volvox, they're kind of cool looking. So what we tend to do is we have 2 things for you to look if you were in the lab. One, we would have a live culture, which I don't know if I've ever gotten a student to find one of these. They're really hard to find in the media. But we do have pre-made slides, and they're stained. And so with those, you can see the nice nucleus. So if I ask you if this cell is prokaryotic or eukaryotic, you should clearly be able to tell me that it is eukaryotic, because it has a very clear nucleus. All right, after I got rid of that dog. So it has a really clear nucleus here, and you can also kind of see the other organelles inside there. And they move really slow. You may have heard of the brain-eating amoebas. If you haven't, they're kind of fascinating. They like to, they tend to repopulate ponds and lakes when the water gets really warm, and they tend to sit on the bottom. But when people are swimming and moving around, and when sometimes we have the spring and fall, what we call turnovers, where the water columns flip, they get into the water, and then people go swimming, and usually what happens is it goes up your nose while you're swimming and then gets into your brain, and in most cases it's fatal. So if you have heard of the brain-eating amoeba, that is true, but not all amoebas are bad. Then we have the paramecium. Now the paramecium, usually when we get these out of the live culture, and a lot of times we see them when we look at the Elodea, the aquarium plants too. So again, you can see there's nice organelles in there. This would be what the live one looked like. Now we would have had a nice one. This one's stained. They are already prepared and fixed, so they're dead, right? So we squished them in there, glued them to the slides already pre-made for you, and you can clearly see the nucleus inside this cell. So that can definitely stand out as being eukaryotic versus prokaryotic if we ask you that question. The thing with the paramecium is they do tend to move very fast. So we have a solution called ProtoSlow, and what ProtoSlow does is, instead of putting water on your slide, you'd put the ProtoSlow or add the ProtoSlow to the water, and it's really thick. It would be like if you were to fill a swimming pool with caro syrup, if you know what that is, or molasses. Be hard to swim through it because it's so thick. And so that's what we do. And so they move slower so you can get a better view of these specimens. And then that is the last lab for lab 3. So now what you need to do is you need to go through the review. When you go through the review, you can send me a copy. And then just like I said, take a picture, upload it, send it to me. You should be in the habit of doing this by now. And then don't forget your 5-point assignment for, again, just take your screenshot and attach that to the assignment and tell me your approximate size. So hopefully I was able to answer all of your questions, and again, watch the videos, and good luck with lab 3.